Hi, this is Ikuo Tsunoda, Assistant Professor at Louisiana State University, Shreveport, Louisiana. This picture is the one that when I attended a fundraising event, Walk MS in Salt Lake City, Utah. Now let's move on to the second part. Is MS virus infection? The viral hypothesis of MS is that direct virus infection or antivirus immune response causes demyelination. This is supported by the facts that viruses themselves and antiviral immune responses have been detected in MS patients. Herpes virus has been associated with MS. Rhinovirus, which causes common cold, is also associated with MS. These rhinovirus belong to the picornavirus family like poliovirus. The second evidence is that some viruses are known to induce demyelination in animals. The most frequently used viral model is Theiler's virus infection. Theiler's murine encephalomyelitis virus, TMEV, is a non-enveloped single-stranded RNA virus and belongs to the picornavirus family. Other viruses that belong to the picornavirus family are poliovirus, Coxsackie virus, Ikuo virus, or rhinovirus. Theiler's virus was first isolated by Max Theiler in 1934. Max Theiler is famous for development of vaccine against yellow fever, and he was awarded the Nobel Prize in 1951. The disease caused by Theiler's virus is similar to MS, both clinically and pathologically. Clinically, infection in susceptible mice, such as SJL mice, results in paralysis and the disease course is chronic progressive. In contrast, resistant mice such as C57BL6 and bulb C mice can clear virus. Pathologically, in susceptible mice, you can see inflammation and demyelination with virus persistence. As I will explain in the next few slides, axonal degeneration is also seen. The cause of demyelination is not clear. There are two theories. The first one is that direct virus infection kills myelin-forming cells, oligodendrocytes, and causes demyelination. The second one is that demyelination is mediated by immune responses. Some groups believe CD4 positive T helper 1 cell is an effector, and others believe that CD8 positive cytotoxic T cells is the effector. Antiviral antibody has also been shown to cross-react with myelin components and induce demyelination. Theiler's virus causes a biphasic disease in susceptible mice. 1 week after intracerebral infection of virus, Theiler's virus infects neurons and causes acute polioencephalomyelitis. This is immunohistochemistry against the virus. You can see virus antigens not only in cell bodies but also in processes of neurons. The virus spread in the CNS using axonal transport. And 1 month later, the mice develop a chronic progressive demyelinating disease and show paralysis. During this chronic phase, the virus persists in the spinal cord. This is how we process the spinal cord of mice. Spinal cord was cut transversely. If you see the section of the spinal cord, butterfly-shaped gray matter exists inside, and it is rich in cell body of neurons. The white matter locates outside, and here is rich in myelin and axons. This is a time course study of myelin staining of the spinal cord in Theiler's virus infection. In this luxofast blue staining, myelin sheaths are stained blue. This is the anterior horn of the gray matter, and this area is the white matter, which is rich in myelin. As I told you, obvious demyelination is first detected 1 month after infection. These white vacuolar areas are demyelination. You can also see inflammation around vessels in the white matter 1 month after infection. In contrast, during the first 3 weeks after infection, most areas of the white matter appear normal, and only mild cell infiltration can be detectable in some areas. However, in the next few slides, I will show you many damaged axons in these normal appearing white matter. We detected axonal damage using immunohistochemistry against neurofilament. Neurofilaments are phosphorylated in the cell body of neurons and are transported to axons. So in normal neurons, the non-phosphorylated form is detected in the cell body, and the phosphorylated form is detected in axons. When neurons and axons are damaged, you can see the opposite distribution of phosphorylation. The phosphorylated neurofilament in the cell body, and the non-phosphorylated form in axons. In the next slide, I will show you staining using this non-phosphorylated neurofilament staining. 1 week after infection, most area of the white matter showed no staining. However, you can see axonal swelling positive for non-phosphorylated neurofilament in the lateral funiculus and the ventral root exit zone. In higher magnification, you can see retraction balls, an indicator of axonal damage. The number of damaged axons increased over time. You can see beaded and fragmented axons. As I showed you two slides ago, this area of spinal cord appeared normal by conventional myelin staining. Since obvious demyelination was detectable 1 month after infection, these results suggest that axonal damage precedes demyelination in Theiler's virus infection. Next, we examined whether damaged axons co-localize with demyelination at a single fiber level using confocal microscopy. We visualized the damaged axons shown in green with myelin sheaths positive for myelin basic protein MBP shown in red 2 weeks after infection. A merged image shows distended damaged axons wrapped with myelin sheaths in the white matter. This shows that axonal damage precedes demyelination. In this slide, you can see a virus-infected oligodendrocyte in red and normal axons in green, stained by phosphorylated neurofilament. Here, one single oligodendrocyte wraps multiple normal axons. You can see red myelin sheaths outside and green axons inside. Interestingly, the top myelin sheath, pointed by a large arrow, lacks normal axon stain and looks empty. Let me explain again. This is one single oligodendrocyte that wraps several axons. The lower 4 myelin sheaths contain axons, but the top one does not contain the axon. This suggests that the axon is either damaged or lost before myelin destruction. Therefore, this is another supportive evidence that axonal loss or damage precedes demyelination. In the next slide, I will show you this empty myelin profile in multiple sclerosis. This figure is from a case report of an MS patient. Myelin sheaths are shown in green, and axons are shown in red. These normal myelin sheaths contain axons, but those myelin sheaths contain no axons. These results suggest that in human patients with MS, axonal damage can also precede demyelination. In Theiler's virus infection, we also looked at how macrophage and microglia in the spinal cord respond to axonal damage. 2 weeks after infection, we can see activation of microglia that have dendritic processes. 3 weeks after infection, microglia and macrophages change their morphology. These microglia and macrophages seem to take up degenerated axons and became rounded phagocytes. The number of reactive microglia macrophages increased over time. Based on the results, I have proposed a new model for lesion development in Theiler's virus infection, the inside-out model. First, axonal damage occurs inside, then demyelination follows. Here, the lesion develops from the inside to the outside. I just proposed that there are two hypotheses how axons are damaged in MS. The outside-in model for autoimmune disease EAE, and the inside-out model for Theiler's virus infection. However, I would like to emphasize that these two hypotheses are not mutually exclusive. For example, in EAE, first, lesions develop from the outside in. Anti-myelin autoimmune responses initially attack myelin, and axons are damaged secondarily by severe inflammation. Then, if axons are further damaged and severed, the distal stump of axons will degenerate. This is Wallerian degeneration. From here, the myelin sheath wrapping degenerated axons will also degenerate. At this stage, the lesion develops from the inside axon to the outside myelin. Then, this degenerated myelin will be taken up by microglia and macrophages, and myelin antigen can be presented to autoimmune cells in the brain, and the activated autoimmune cells can attack myelin from the outside. This will lead to the second cascade reaction and disease progression. If this is true, this vicious cycle can occur regardless of whether lesions develop initially from the inside or the outside. As you see, the key event in this vicious cycle is Wallerian degeneration. So we asked if there is no Wallerian axonal degeneration here, what happens? This is the end of the second part.